Okay. All right. All right. Spend 2 minutes. Okay, everyone, let's come on out. Let's get ready to start this. I'm super excited. I'm super pumped. This is amazing. It's an amazing turnout. This is the time for biology. Biology is having its moment, and I couldn't be more thrilled to be a part of that. So thank you all for braving traffic, for taking time out of your day, and for coming to see these amazing teams and what they've accomplished in the last 100 days. It's actually quite phenomenal what these guys have done with a lot of hard work and a lot of sweat and some tears. So it's great, and thank you again for sharing that with us today. So before we get to the stars of the show, the teams, I wanted to spend like a couple minutes talking about what IndieBio is and why we're doing it. So biology is actually accelerating now faster than Moore's Law. Many of you guys have seen the Carlson curve. This is the cost of DNA sequencing. It's falling rapidly. Ever since 2007, continues to fall rapidly. But along with that is not just sequencing, but all aspects of science. DNA synthesis, advances in DNA synthesis, digital PCR. New technologies like CRISPR-Cas9 and DNA editing are promising to make molecular biology much more efficient, faster, and cheaper than ever before. And this is opening the door for independent biology. It's what allows us to build IndieBio. And this is great timing for a lot of the academics out there, PhDs, students that are taking a lot of time to go through school, earn their PhDs, and then find out about the postdoc-opocalypse. I don't know how many of you have heard of that, but that is a term that's been coined by postdocs about how there's too many postdocs for the number of tenured faculty positions. And so they end up going to big pharma or industry and find out that they're working on things that may not be what they wanted to work on, or may not employ their full creativity. So the door is now opening to independent biology at a perfect time. These scientists can now become entrepreneurs with help and a little bit of seed funding to de-risk a product or a technology and turn that into a real business. That's really exciting. So they're doing that with the help of IndieBio and others that will turn what their ideas are into these technology or into these real businesses through business development, product development, actual science development, where we're using parallel iteration, using other techniques to actually speed up science in the lab as well. And so the whole cycle looks a lot more like building a tech company than an old school biotech company. Fundamentally, we also want to do this in a way that allows founders to retain control of their companies. And we're doing this because we want scientists to be working on the world's most intractable problems. Here you see population growth through history. And you can see a massive spike around 1800 or so. And there is no reason that this isn't going to continue to climb. As technologies come online, as our life expectancy continues to grow, we're going to have enormous pressures on the resources of our planet. We're going to need these scientists and working on these intractable problems to solve for these issues, like extinction, drought, famine, disease, and discovery. So we exist purely to help these scientists solve these huge problems. Super exciting for me, and it takes a whole team. So I'd like to introduce Ryan Betancourt. He's our program director. You probably all know him, I think. And this is Ron Chigueta, our chief science officer. And together, we all work every single day side by side with the teams and helping them turn these ideas and these basic technologies into real businesses. And to get a little bit more flavor for that, I'll let Ryan talk a little more.
Great, thank you, Arvind. We'll just switch over to my one slide. So coffee's for closers. And what do I mean by that? I mean that great companies are built by founders who close deals. So that's really what we focus on at IndieBio on the business side. We focus on helping founders take their technologies and turning them into businesses. We do that in 3 key ways. We do that first by focusing on the product. We look at their product and what they're trying to develop, look for product market fit with them, and we go through the entire process. So as an example, one of our companies, through the process of interacting with potential customers, found that there was a huge need for their service with next generation genomic diagnostic startup companies. As a result, they signed several partnerships, they put several partnerships into place, and they're moving their business forward. The second is sales. We believe sales solves everything, particularly for small, growing companies. One of our companies came in, and you'll be hearing from them a lot more shortly. They developed a product in-house. They needed to build a sales strategy. We built it with them. They developed a sales funnel, and then they started to go out and close deals. They were actually the first company at IndieBio to start pulling in revenue and customers. So this is a biotech company that makes money as of 4 weeks ago. The third, and equally as important for us, is survival. So we focus on ensuring that our founders and the companies they build survive. This isn't about IndieBio and the accelerator. This is actually about building high-quality businesses that survive over a long period of time and that are sustainable. One of our companies, which again you'll be hearing about very shortly, built their minimum viable product, launched, and a couple of weeks ago, after a couple of weeks, they have users in 150 different cities globally. So this is just to give you a taste of some of the companies that are going to be presenting today. We're all incredibly incredibly excited by everything they've achieved so far. And with that, I'm delighted now to introduce you to Dr. Ron Shigeta, our maker of all things. Ron can build wet biology, software, and hardware. Ron?
Thanks, Ryan. So we can't really accelerate a biotech company without accelerating the tech. So I want to tell you a little bit about the company's experience in the last 3.5 months. So we have a lab that's the center of the program, 2,500 square feet of lab space for molecular and cell biology, as well as a little chemistry and some hardware as well. And while that's really useful, and we do encourage the companies to take high iteration, highly parallel strategies, find backup plans, one of the things that's really been exciting for the past few months is seeing that every company is actually helping to accelerate the other companies. The class itself are one of the key ingredients to how the accelerator works. Molecular biologists came in, sat down on the first day, and started working right away. But then they would find that they're sitting next to an experienced product chemist, or next to someone who really understands machine learning. And all these relationships have been very, very helpful as the companies encounter their obstacles. Every technical plan that you see today had major shifts in the last 3.5 months, and they've all overcome those problems. Another big ingredient to accelerating tech has been our network of mentors, who we've been really, really pleased to work with over the class. And they have domain knowledge, and also sometimes very, very senior in experience. So the technical plan discussion here of CSOs on the panel here, every one of these individuals on the panel have taken a product to market for more than $10 million in revenue a year. That sort of level of product has added decades of experience to every company's capabilities and really helped get the results that we're seeing today. The teams themselves have had some amazing results, and so all I can say is that 7 of the 12 teams that you receive today, they came with paper-only plans. They had no actual work on the products you're about to see. But today you'll see every single one of them has a product. All right, so I guess it's time to get started, and so I'm going to bring up our first CEO. Art, you want to come up and tell us about Clara Foods? Thanks, guys.
I'm Arturo, and I'm with Clara Foods, and we're creating the world's first animal-free egg white. Egg whites, as many of you can tell, have become incredibly popular over the last few years, making their way into grocery stores and restaurants across the country. So much so that they've tripled in price for the last 3 years to command over a $3 billion market in the US alone. This surge in demand, however, comes at a greater cost to the environment, the animals, and our wallets. And it's precisely the problem that brought our team together. Dave saw this as an enticing technical challenge. I saw it as an incredible business opportunity. And Isha saw it as a way to make our food system far more sustainable. And the more we thought about egg whites, the more we began seeing them as a product that we could make ourselves. And a surprisingly simple one at that. Egg whites are 90% water, 10% protein. They have virtually no fats, no cholesterol, and no carbs. And over 90% of the protein content is represented in just 7 proteins. These 7 proteins are also what give egg whites their incredibly diverse set of functionalities, ranging from gelation to foaming, emulsifying, and clarifying. And we've discovered that by varying the protein profile, not only can you create an egg white that cooks, looks, and tastes like its hand-derived equivalent, but you can create new kinds of egg whites that actually outperform their hand-derived equivalents. For example, we've discovered that some proteins antagonize each other for certain functionalities, and by removing them all together, we've created new egg white formulations that outperform their hand-derived equivalents and foam with more stability and use far fewer inputs. It could translate into new kinds of foods. Imagine the fluffiest angel food cake you've ever had, or an extra light meringue like this one, or these that taste like heaven. But working at the individual protein level to make all these possibilities a reality means using an incredibly inefficient and outdated factory we call the chicken that converts resource-intensive inputs into proteins. We have to feed it all this water, grain, pump it with antibiotics and hormones, wait 20 to 25 weeks for it to start laying eggs, get the eggs, separate them, sometimes even throwing away the yolk, and then get the egg white and reverse engineer it to get to the individual proteins. We use nature's far more environmentally friendly and efficient factory called yeast. And in the same way that brewers use yeast to convert sugar into alcohol to make beer or wine, our yeast is specialized to convert that same sugar water into egg white proteins. Our process allows us to create egg whites that are inherently superior without compromising on the qualities that we know and love, and still be able to deliver on these new performance capabilities. With all these advantages, we are uniquely equipped to work with some of the largest egg white users. And I'll use Barilla as an example of a potential customer. For starters, we can work with companies to help them meet their sustainability initiatives seamlessly and painlessly without compromising costs or quality. We can work with them to create new products and make new egg white formulations that are tailor-made to their needs. In Barilla's case, it may be an egg white that has a higher protein profile without compromising the binding abilities that are so essential to pasta making. But I like to think that the greatest value that we can deliver to our corporate partners is de-risking their supply chain in ways that animal products cannot. Because we're decoupled from the chicken, we can match demand perfectly, lock down prices for an extended period of time, and have no risk of recalls due to salmonella or any avian-borne diseases. But not only can we compete on the basis of quality, we can also compete on the basis of cost. Our process allows us to create our egg whites and undercut even the wholesale market. And these prices of $20 a kilogram that I just mentioned, because of the avian flu that's all over the Midwest, prices as of 2 weeks ago were close to $50 for wholesale dried egg whites. And we're fortunate that we can exploit several revenue streams as we build a sustainable business. And that means that we can start selling the individual proteins because they have commercial value as standalone products, which we are MVPs. And then we can start selling the specialty formulations that use far fewer proteins, but still can deliver on those performance capabilities and we're therefore able to capture the upside margin. This is all on the road to creating our mass market standalone B2B product, Clara Whites, which will allow us to make hen egg whites a thing of the past. We're fortunate and excited to announce that we've raised the $1.7 million that will allow us to get to small-scale production and deliver our initial batches to our corporate partners and have orders in place in time for our Series A next year. Thank you very much.
Hi everyone. Wow, I have to keep up now with Clara Foods. Hello everyone, my name is Jaime. I'm the CEO and co-founder of Auryn BioCloud, and we're a genetics lab in the cloud. Until recently, doing genetic engineering with something only affordable for big pharmas or big labs. Why? Well, I'll give you two compelling reasons for this. The first, that you need high technical skills to do experiments, especially in genetic engineering. Also, you need a lot of money to buy expensive equipment and resources. And this is keeping really brilliant minds out of this industry. Also, if you want to go from idea to actual execution and results, there's so many steps involved in this process, and it takes time. So for example, you have to first synthesize. Then you have to go and buy the different reagents and cells from different providers to then take it to your lab or someone else's lab and build it. And this step is not once, but several times because you start tweaking it. This takes a lot of time and is very tedious. And that's why we built Auryn BioCloud. It's a genetic engineering platform that allows users to go from concept to execution in a non-stop, easy, and fast way. So we've built a virtual lab that provides you with all the resources that you need to physically carry out your experiment. It doesn't matter where you are in the world or what background you bring in biology. And this is not skipping the amazing biotech companies out there. It's providing a layer on top so we can make it even simpler for our users. So let me show you how it works. It's not magic, it actually works. So let me show you. This is our user interface. You create very quickly a project by naming it, putting a description, and then choosing out the different DNA fragments you want to put in it. Very, very simple. And then when you're happy, click assemble, and it gets done. Let me show you a project of one of our users, a very early user. And this is how it looks, like a timeline. You use Facebook, this is familiar. And what you do, you see every single step, even a live stream that shows you how the experiment is actually getting carried out. This user, by the way, was actually a software developer. And here I have a copy of his construct. And what he did is he reprogrammed this bacteria so it would glow in the dark. Simple, but he did it. And to support this platform, we had to build our first machine, what we call it a bioserver, a machine that contains all the units and the resources required to carry out gene assembly, transformation, plating, and incubating, all in a completely automated way. So let me explain you a little bit more of our vision. We see putting more of these machines together so people can carry out multiple experiments so they can come back to results very quickly. This is the power of parallel processing. And that's why we started building our second machine, which is going to be bigger, better, and way faster. So let me explain you how this actually works. We have a library of different DNA fragments that you can use. These parts were actually created by our community. And you can just put them together with the visual interface you saw, and it gets physically carried out in our labs in San Francisco, meeting all regulations in the United States. And what's even more simple is our business model. Because what we've done is bring down the cost of a basic construction just to $80. That's even lower than what you can find in the market, which usually goes around $300. And if you want to add additional parts, let's say another promoter, another gene, it's simple. You pay an extra $80, which still keeps you under the market value. And, of course, without having to own a lab or all these machines. So we are live already a month and a half ago, and we started to see a lot of projects being built. Actually, today, while demoing, we saw a user create a new one. So this is pretty good. And we started to see more advanced projects that we're working with. For example, creating more affordable ways to produce insulin or drugs that may treat for cancer. So imagine all the thousands of projects that can be built on our platform. We already started working, we're negotiating with the University of Colorado to provide this platform for their students. And they have a budget of $250 million just for research in genetic engineering. So this is really valuable for them. But what was interesting is that we started to get a lot of attention actually from individuals. Without doing any marketing, people are starting to get into the platform and running their experiments. These users are not biologists. Most of them are students, software developers, which shows that it's a demand across different backgrounds. So we started to create this community because we believe this will be the next big market. That's why we've built a forum where these brilliant people are already creating their new projects and working together, something that we usually don't see. And we have today over 370 users registered, ready to run their experiments. And what's even more interesting, they come from 165 different cities around the world, which really shows the international demand this platform has. But what really motivates us even more is feedback, and especially from our paying customers, which shows that we are in the right track. And one of them is the founding scientist of 23andMe. And what he says we do better than anything out there is that we give speed of executing projects. It's simple, the approach we've taken, and it's way affordable than anyone else. So this is what motivates us to continue doing this quality. We have, just last week, more than 3,000 visitors, which shows the active use of our platform. People working together to create the next thing in biotech. So, with that said, we're looking to raise today $1.5 million to scale, to expand the team and put more machines on the platform. Last but not least, I wish to introduce you to my co-founders. We're the team behind Arcturus BioCloud. And if put together, we've done 8 companies in the past with a combined revenue of $10 million across different industries. We have 3 former CEOs, 2 former university professors, and one of them is a PhD with more than 12 years of experience in genetic engineering. We also come from many prestigious institutions around the world. Even Singularity and their crazy ideas about the future. But we have the domain expertise required to address this challenge. So, we invite you today to invest in Arcturus BioCloud. Thank you very much.
Rhinoceroses are vanishing. They're being poached from Africa for an ancient tradition, a tradition recorded by Li Shizhen. Li was a pharmacist who lived in China in the 1500s. He wrote about the uses of rhinoceros horn in traditional Chinese medicine. Back then, it was a fever reducer. Today, it's a cure-all, an aphrodisiac, and a status symbol. People will risk everything for it. That includes the poachers who are willing to die to acquire it, and the end users who face imprisonment for using it. Something needs to be done. Oops. We, George, an expert in keratin, Rebecca, a tissue engineer, Daniel, a practitioner of traditional Chinese medicine, and myself, Matthew, a 3-time entrepreneur, came together with the belief that animals are precious and traditions are important. Here's what we learned. You can source keratins from natural and synthetic sources. You can then run them through a series of chemical reactions to construct a keratin matrix that entraps the other components of rhinoceros horn. Finally, you can add rhinoceros DNA to the mix. The end result is the world's first synthetic rhinoceros horn powder. This powder, when spectrographically analyzed, matches rhinoceros horn. It also yields the fingerprint of rhinoceros when genetically tested. And it can be used as an ink in a 3D printing process to produce horns like these right here. The illegal trade in rhinoceros horn consumed a record 1,215 animals last year. That's over 6,000 kilograms of horn. With a black market price of $60,000 per kilogram, the total market size is $437 million. Now, it's been estimated that the latent demand for rhinoceros horn is over 300,000 kilograms of horn per year. Pembient can meet that demand at 1/8 the black market price. Thus, we have the opportunity of servicing a $2.6 billion market by 2022. Furthermore, by dropping the price, we'll make money, but the poaching syndicates won't. If we sell just 30,000 kilograms of horn at the new price, we'll generate $288 million in revenue. With a 74% gross margin, that's $168 million in gross profits. Subtracting out expenses, our earnings before interest and taxes will be $88 million. So with a price to EBIT ratio of 11.5, Pembient will have rhinoceros horns, but it'll also be sporting a unicorn horn. Our go-to-market strategy is to enter the market as a branded ingredient. We're going to be just like just like Gore-Tex, which is North Face, we'll be in a variety of consumer brands. Here are some of our branding partnerships to date. We developed a skincare line that we're in talks to spin off. We're working with the 3rd largest craft brewery in Beijing to brew a rhino beer. We're also looking for partners in the tobacco and durable goods segments, and we're in talks with a pharmaceutical company in Vietnam. Now, I'd like to show you a video produced by a Vietnamese director around our skincare line.
Bí quyết của sắc đẹp là gì? Niềm tin. Niềm tin có thể xóa đau thương. Niềm tin có thể hủy hoại bạn. Từ ngàn năm, tổ tiên đã tin rằng sừng tê giác mang lại sức mạnh vô biên. Thế nhưng, thế giới của chúng ta dần bị ô nhiễm. Sừng tê giác cũng vậy, và nguồn gốc của chúng là một câu đố. Niềm tin cần phải được phát triển như sự tiến hóa của chúng ta từ quá khứ đến tương lai. Vì vậy, tôi sử dụng Essence Rhino Horn, một sản phẩm tinh khiết, an toàn, dễ thích ứng với cơ thể và là một sản phẩm mang tính nhân đạo hơn những chiếc sừng tê giác đang được lưu hành bất hợp pháp. Essence Rhino Horn không đến từ Trung Quốc hay châu Phi mà đến từ vùng đất nguyên sơ tại Tây Bắc Thái Bình Dương ở Hoa Kỳ, nơi các chuyên gia khoa học đã ứng dụng công nghệ sinh học và tái tạo lại được sự kỳ ảo của sừng tê giác cho làn da mềm mại và tươi mát hơn. Nắm bắt tương lai, dưỡng chất tin cậy.
I'll let them know everybody applauded. Thank you. Our research indicates that Pembient horn has a perceived authenticity and is much better than water buffalo horn, the current substitute for rhinoceros horn. Additionally, Pembient horn is built in a lab, so it's pure. It contains none of the pollutants found in wild or farmed animals. Here are some of our milestones. So in 2014, we started this project and produced some synthetic rhinoceros horn powder. We took that powder to Vietnam and did customer development. Upon return, we acquired some wild horns from a museum and continued our R&D process. We were able to print crude horns at the beginning of 2015, and now we're looking forward to our initial product launch in Beijing in the fall of this year. Beyond that, we'd like to expand to other wildlife products, including elephant ivory and tiger bone. We're raising $1 billion, and currently 60% of our round is already reserved. We're Pembient, bioengineering rhino horns to fight poaching. And one more thing. Today, Pembient, in partnership with the University of Washington and New Harvest, is announcing the Black Rhino Genome Sequencing Project. We're crowdfunding this through experiment.com. So if you're interested in advancing science, please go to experiment.com and donate. Thank you.
Hi everyone, my name is Kathy and I'm the co-founder and CEO of Renomics. To me, genetic tests are all about answers. Since the discovery of the genetic code, the most valuable way we have been using that knowledge is through genetic testing. Genetic tests are able to tell us our likelihood of getting a tumor or heart disease or just about any hereditary disease. And this has become a vital part of preventative medicine today. However, there's a secret in the industry that these genetic testing companies know and is hiding from the public, and I'm about to share it with you today. The secret is that more than half of your genetic testing result consists of data that they cannot understand. What I mean by that is, for example, let's take BRCA1, the most commonly screened gene for prospective breast cancer patients. In this one BRCA1 gene, there are 1,863 amino acids. And since there are 20 different types of amino acids, there are about 37,000 variations just for this one BRCA1 gene. Now, how much do we actually understand about these variations of disease genes? And the answer is very little. These genetic testing companies only understand about 1% of all 37,000 variations for BRCA1. And that leaves about 36,000 variations left unknown. And this problem is that most of the disease-causing variants lie within that 99% of unknown variants. So this makes genetic testing very inaccurate and very inconclusive. So in a more clinical setting, we saw the case with Angelina Jolie last year. So she got a double mastectomy because there were variants found in her BRCA1 gene that were deemed to be pathogenic. But now that we know that 99% of the variations of these hereditary disease genes are actually unknown, this poses a question. Did she make the right decision? But more importantly, do we want this uncertainty for the rest of the world? And even with so much lacking information, this is still a very big industry. And it's growing at a rate of 10% every single year, and it's expected to be $25 billion by 2021 in the US alone. So this inspired us at Renomics to come up with a radically different approach to this problem. At Renomics, we can actually understand 100% of all the variants. So we can understand all 37,000 variations for BRCA1. And the way we do it is that instead of waiting to see that variant in a patient, we actually get 37,000 variations and all the variations for any human disease gene. And we can, on a high throughput manner, we can look at all these variations in disease model organisms and see how pathogenic, how bad each variant actually is. And this is all carried out in a wet lab setting. And we compile all of this information in the Renomics database. So the next time when a genetic testing company sees this variant of unknown significance, they would just query our database. So the first product we'll be developing is called Variant Onco. It consists of 20 of the most commonly tested hereditary cancer genes. And these genetic testing companies will be charged a fee per query to access our database, essentially. And in the last 3 months of IndieBio, we've already been told that variants of unknown significance is a huge problem for every company out there. And this is what it would look like from the user's perspective. So they would search the gene name, the variant ID, and they would get a variant analysis report based on the experiments that we've already conducted. And it shows whether or not that variant is pathogenic or it's not pathogenic. And now going to the economics of this first product we're developing called Variant Onco, we expect to break even within less than a year of releasing this product by just capturing 10% of the genetic tests performed by one single company. And just by adding one additional customer every year, we expect to reach a yearly revenue of $53 million by 2021 for this one product alone. Now, in the last 3 months of IndieBio, not only have we talked to customers, but we've also validated our scientific pipeline. We've actually manufactured these variants in the BRCA1 gene, and we compared it to clinical data, so data that's already been seen in patients, and it matched up perfectly. So now we're at the stage where we want to scale our technical pipeline to 20 more genes to create the variant ENCODE database within 12 months. And after tackling the hereditary cancer space, we're going to the prenatal testing space, as that's the second largest submarket in the entire genetic testing industry. And by 2020, we expect to be the world's best database for functional variants for all hereditary disease genes. And this is our founding team. So Leo and I are both from Toronto. Leo is finishing up his PhD at the University of Toronto in molecular genetics. We also have a great board of advisors, including Dr. Fritz Roth, who is a pioneer in East Genomics, and Matt Fikowski, who is the founder of Invitae, one of the largest genetic testing companies in the US. So now we're raising $1.5 million to take us to our next milestone. And 1/3 of our round is already committed. Thank you.
Good afternoon, everybody. I'm Herschel Chokhawala, the CEO and co-founder of ZymoChem. At ZymoChem, we are developing the next generation of microbes to make industrial chemicals. The world that we live in today is built from petroleum. From the paints in these walls, to the carpet on the floor, to the chairs that you guys are sitting on, everything is made from chemicals that typically come from petroleum. And the global petrochemical industry is a $3 trillion a year industry and growing. But to feed our dependency on oil, we do crazy things, like building rigs in the middle of the ocean and then digging all the way down in the ocean floor just to get oil. It's unfortunate, but we do this because till date, we haven't had any other alternatives to fulfill our needs. But now we do. Advances in biology have spawned many companies that can now engineer microbes to make these exact same chemicals, but from something that is as abundant and renewable as sugars, using processes that are as benign as brewing beer. Almost everything that we use today should be made this way. Unfortunately, it's not, because brewing chemicals is still more costly than making it from petroleum. Why is that? Well, when microbes convert sugars to the desired chemical of choice, they lose at least 1/3 of the carbon in the form of CO2. Think the foam on the top of your beer. Now, because of this carbon loss, you make much less product than you would otherwise. Also, sugar is the single most expensive component of the entire process, and this loss translates to 20% of the total cost, which effectively kills the economic competitiveness of brewing chemicals. So when I looked at this and I thought, well, there's got to be a better way, right? So at ZymoChem, we decided to kick around the idea of designing a microbe that would take all the carbon in the sugar and just make the chemicals and no carbon dioxide, right? So this was an idea. So if you're successful in doing this, what this means is that you can get up to 50% more chemical than state of the art today. And for many chemicals, that means that you can brew chemicals profitably, even at today's oil prices, if you can do this successfully. So I'm sure you're all wondering, how do you do this, right? What's the magic? So when microbe makes chemicals, they first convert sugar to a universal intermediate, which is called pyruvate. Existing technologies convert pyruvate to chemicals using a path, wherein the very first step is the one that is responsible for this carbon loss. This is a naturally occurring step, and none of these technologies can function without this step. And because of this, they suffer with the carbon loss. So to avoid this at ZymoChem, what we had to do is design a completely new path, one that does not exist in nature, to make chemicals from pyruvate without losing any carbon as CO2. Each individual step of our design pathway is designed that it would not lose any carbon. And because of that, we can now convert carbon all the way from sugars to pyruvate to your desired chemical of choice, giving us better yields and better economics. I'm very happy to tell you that we have not only patented this under the PCT, but also assembled a kick-ass team with more than 3 decades of experience at Cal, Stanford, and Imperial College London that is executing this technology today. Hi, once again, I'm the CEO and the inventor of the technology. My expertise is in discovering enzymes that catalyze each individual step that is unique for our technology to make chemicals. My co-founder in CSO, John Kuchenreiter, is the builder of the microbe. He spent much of his life at Cal and Stanford building microbes to make chemicals, something that he's very used to. Our third co-founder is Murthy Konda. He's the economist. Yes, economist. Why? Because for us, economics make a lot of importance and it's very critical. So even before we actually go ahead and do a single experiment, we figure out, using the help of computational models, whether the chemical that we want to make with our technology can be profitable at scale, given the constraints of making it from petroleum. Guiding us in our efforts are stalwarts and eminent scientists in this field from UC Berkeley as well as MIT, and also industrial leaders who have helped the Generation 1 companies scale up. Also funding us is the government for making 2 products, 1-hexanol and adipic acid, using our technology. Both these products go into everyday products such as lubricants as well as perfumes. And adipic acid is the precursor for making nylon 6,6, which goes into making ski jackets as well as the carpet that we are all standing on. For 1-hexanol, we have already validated enzymes that catalyze each individual step for making it from pyruvate. This is a significant milestone for us because it was a big unknown a year ago. Our second milestone, which is basically putting all these enzymes together in a single pot to make 1-hexanol. And we anticipate finishing that by the end of this month. Once we do that, this will unlock a phase 2 grant from the government for $500,000. Similarly, for adipic acid, we have also validated all enzymes that would make this chemical from pyruvate, completing milestone 1 for this grant. The second milestone involves building a microbe around this technology, which we anticipate completing by the end of this year. Once we achieve that, it will unlock a phase 2 grant for $1 million from the Department of Energy. Now, based on our progress and the potential of our technology to actually allow brewing these chemicals profitably, we have received letters of intent from the Nylon Corporation of America, as well as a big Fortune 500 company to make both these chemicals at scale for them. And to get there, for just one product, adipic acid, we anticipate 3 years of work to scale our technology all the way up to demo scale, where we hope to be competitive with petroleum-based adipic acid at the price of $1.70 in oil. Today, we are announcing a fundraising round for $750,000 that, in addition with the non-dilutive funding that we have received so far, would be used for the microbe production of both these chemicals. We are Team ZymoChem, and we want to make sure that the next nylon jacket you guys wear comes from sugars and not oil. Thank you.
Hi. My name is Francesco. I'm with Sensa IO, and I make bioreactors. So, what is a bioreactor? It's a controlled environment for growing microorganisms, mainly for their byproducts. And why should you care? Well, probably you already do. Everything on this list is the end result of microorganisms just doing what they do. And human culture owes a lot to them. Beer, for instance, is likely as old as civilization itself. It's not very difficult to make. You take grains, you soak them in water, you add the specific microorganism, in this case a yeast, and it converts those grains into alcohol. Everything on the left side of this list is relatively easy to make like that. But the things on the right require a bit more of a refined approach. So we build these ridiculous-looking machines called bioreactors because we need to control what's going on inside and protect it from the outside world. But bioreactors today have a number of drawbacks. First, they are expensive, thousands, if not tens of thousands of dollars. Next, they're locked down in ways which generally restrict scientists and what they can do. Maybe restricting what the user can do for an iPad sounds good, but when you're trying to control and harness the power of another living organism, you need all of the control you can get. And finally, good luck checking on your brew from your mobile phone. But we at Sensa IO are trying to change all of that. Ron, if you would. Scroll this over here. We're building a low-cost, open solution, a platform that doesn't restrict scientists by design. Open source hardware and software means that ultimately it's the community that decides what this device can do, not me, not any specific company. So whether you're brewing kombucha or brewing yeast, the steps should be as simple as load in the ingredients, download a recipe, and push a button. So currently, we're working with Real Vegan Cheese. They're a project right here in Oakland, and they're making cheese without cows. They're helping us test and debug our technology, and once we're done, once we have good results, other companies presenting here today are interested, as you might expect. That's a little fast. The recipe is not quite right. So let me just lay out for you why this is important. Why do we need a flexible, open solution? Well, I hope the other companies presenting here today are convincing you that the way we produce food and drugs and other chemicals is going to change massively, is changing massively right now. And that's going to have tremendous benefits for both the individual and the planet. But basic production is a huge barrier. It is, and we need to knock this barrier down. Let me just give you the example of egg whites, for instance, Clara Foods. Right now, the egg in your pastry or your omelet is produced on a farm and shipped halfway across the state, halfway across the country. But with the yeast that Clara Food is making, we can produce those egg whites right here in this building or at home. Think about what effect that's going to have. Think about what we could do with that. My name is Francesco. I'm with Sensa IO. Please contact me if you'd like to be involved. Thank you.
Hi, my name is Anil, and I'm the CEO of Affinity, and our company is creating a manufacturing platform that can cut the cost of producing biopharmaceutical drugs by 95%. When I'm talking about drugs, I'm not talking about your aspirins or your Liptos, these small molecule compound-based therapies. What I'm talking about are antibodies or antibody therapeutics. These are advanced protein-based therapies that harness your immune system to fight diseases. In recent years, they've been used to fight everything from cancer, arthritis, HIV, AIDS, and quite recently the Ebola virus. The reason we focused on this market is because in the last 20 years, it's grown into a multi-billion-dollar industry. In fact, 1/3 of the pharmaceutical sector's revenue is now expected to arise from immunotherapeutics in the next 10 years. 8 out of 10 best-selling drugs today are immunotherapeutics. These are drugs that generate over $5 billion in revenue per annum. And here's an example of one of those drugs. This is Rituxan. It is a phenomenal drug. It has an 80% success rate of curing ailment. However, the caveat with this drug is that it costs over $50,000 per gram, making it unaffordable for most patients. If this drug was an outlier, that would be fine. However, that isn't the case. Other immunotherapeutic drugs are just as expensive. Avastin costs $54,000. Perjeta, the best breast cancer drug on the planet, costs $72,000. And Solaris, the world's most expensive drug, costs over $500,000. In fact, what this means in real world terms, is that the majority of patients who have these ailments cannot get the best treatments because their doctors or the healthcare providers will not prescribe them to them due to financial reasons. So we asked a simple question is why? Why are these drugs so expensive? And what we did, we dissected the manufacturing platform and discovered there was essentially two phases, the upstream and the downstream. The upstream has seen tremendous cut in cost over the last 20 to 30 years due to advancements in molecular biology and biochemistry. However, the majority of the cost arises in the downstream process. Some academic papers suggest up to 95% of that cost is in this phase. This is the capture and purification stage. So what our company decided to do is look at a technology in the downstream process and try to replace it. So what we created was an affinity column that is synthetic in nature. And because it's synthetic, it's a lot cheaper to produce. Our results, preliminary results, showed that if a stock feed was run through one of our columns, one of these columns, we could capture the antibody that we wanted with the rest of the protein serum actually just flowing directly through. We'll be selling, our business strategy is to sell our columns for about $7,500. This is up to 2.5 times cheaper than the current best technology in the market. These columns also have to be replaced every 50 uses, producing a business model similar to the printer and the ink cartridge industry. We'll also be processing small stock feed for smaller biotechnology firms as well as research institutes. We have quite ambitious plans to take 25% market share in the next 5 years, processing over $1 million worth of antibodies a day. Our current processing capacity is about $10,000 per week. We are currently registered manufacturers with GSK and are in process of doing the same with several global biotech firms, including Roche, Novartis, and Amgen. Our company is looking for a $1.5 million investment to grow our company from its current position and keep up in market demands. I'd like to conclude by introducing our team. Myself from the University of Cambridge with a background in drug development and medicine. My co-founder, Dr. Nitin, who has a background in bioengineering and medical diagnostic devices, and Mike, who's a senior advisor and project manager at Genentech. I'd like to thank you for listening to my presentation, and I welcome you to any further discussions at our booth. Thank you.
Chronic wounds cause over 70,000 people each year to lose a limb. Hi, my name is Jennifer Kames, and here at Biolume, we're healing chronic wounds faster. So raise your hand if you've ever fallen down, had a scrape. That should be like everybody. Okay, so we all fall down sometimes, but the trick in life is to get back up. But sometimes it's harder to get back up. If you have a chronic condition like diabetes, you're at risk for developing a diabetic foot ulcer. 15% of diabetic patients develop diabetic foot ulcers. And not only is it gruesome and painful, but it's costly to treat, and you're wondering, is this gonna cost me my foot? Now, although it's gruesome, there is hope. With advancements in biotechnologies, we're closing wounds faster. One of these advancements is microbial cellulose. Microbial cellulose is long nanofilaments of cellulose excreted from bacteria that come together to form a hydrogel. And this hydrogel promotes a moist healing environment. Cell growth goes up and wound closure goes down. In an open trial, one of our advisors showed that microbial cellulose closes wounds 16 days faster than a petroleum-based hydrogel. That's 16 days where you're not wondering, is this gonna cost me a foot? So, here at Biolume, we have started to make our own hydrogel dressings. I'm wearing one right now. They're easy to apply. We've given them to some of our other advisors. They enjoy them. And they adhere pretty easily. And they don't hurt to take off or on. They also feel like skin, so you're all invited to come over and touch them at our booth. Thank you. Now, we're not gonna just stop at making these hydrogel dressings. See, since this hydrogel is produced from a bacteria, we're engineering the bacteria to produce 3 things. 1, hyperproduction of the material, so the cost goes down. 2, we're engineering the scaffold it produces, so it's easier to use as a scaffold for growing other biomaterials like skin. So we worked with another IndieBio company, and we started an experiment together where we seeded microbial cellulose with stem cells to start growing artificial skin. And then third, we have yet to do this, but we are going to add cross-linking into the scaffold, which will allow for the microbial cellulose to be a better scaffold for drug delivery. And all this is for faster wound closure. Now, because we're tuning two things, we're tuning how the material is produced and then the end product, our tunability is higher than traditional hydrogels. Plus, our production costs are lower because we are taking, quote, sugar water and turning it into this hydrogel. And what is our technology process? So our process is we start with a strain of bacteria. We currently have a patent pending on the metabolic pathway that hyperproduces bacteriocellulose. And then we're working with contract manufacturing organizations, CMOs, to grow and process our material so we can distribute it. And one of our advisors has a medical supply company where he distributes microbial cellulose, and we also have talked to several cosmetic companies who use this not for wound care, but for facial care. And this will get it to the end user who is either in consumers, hospitals, or military. So how does our pricing work? So if we're going B2B, we would sell our material for 10 cents, and if we were developing a brand, we would sell our material for 26 cents. We've done a lot in the 3 months that we're at IndieBio. So we developed customers, we've developed advisors, and we've created a strain that cuts fermentation time down by 50%. So fermentation time is gone from 7 to 3 days. Our next steps is we're going to be working with a contract manufacturing organization to get product out by next year. So the founders, my co-founder is Alex Neckelman. He's overcome a lot to engineer our bacteria. And then myself, a biomedical engineer who falls down a lot but knows the trick in life is getting back up. Thank you, and right now we're looking for partnerships. So if you have any more questions, contact us at our booth. Thank you.
Hi, my name is Raghu Machraju. I'm a co-founder and CEO of A-BioBot. The mission and goal of A-BioBot is to help scientists make breakthroughs by shortening the time for scientific discovery. The breakthroughs in life sciences are not happening fast enough. Why? The fault lies in the essential workflows employed in life sciences. The norm is a slow and iterative development cycle. One starts by choosing a protocol, that's the planning stage. One executes the protocol in the wet lab manually, and then one analyzes the results that come from experiments. There are perils in the execute phase. This particular stage is extremely error-prone and tedious, rendering the whole process irreproducible and non-scalable. Often several iterations are required, and the science is slowed down. Now, ask anybody. If they spend any time in the wet lab, they would agree with what I just said. Now, lab automation and robots help. But once they're acquired, they often end up in graveyards in the basements. Why? Well, you would like robots to be in every lab. You would like them to be pervasive, but they're not, because they're inflexible, hard to use, and blind to errors. Ask anybody who has programmed robots. They would again agree with me. At A-BioBot, we have a solution. We have a secret sauce. And we use machine vision and cameras. And our platforms, our robots, are not blind anymore. They're extremely flexible, and they're very easy to use. And all comes from using the machine vision and cameras. To meet the challenges, we assembled a very capable team. We are all from Ohio State, the Ohio State University. It's important. And Buckeyes. And I'm a professor of computer science, and I'm very well versed in machine vision and machine learning. My colleague Kun Wang is a professor of bioinformatics. Ken Ronaldo is a professor of art and technology, is a well-known robotics engineer. Chaitanya Kulkarni is a talented programmer, and young too, very young. And Trademark is an excellent designer and technologist. So over the last 3 months, we created this desktop prototype, A-BioBot, the friendly and flexible lab assistant, robotic lab assistant. So I'm going to show a video of our product, liquid handling robot, going through its paces to finish master mix for PCR. So this is one of the most commonly used protocols. So the scientist is setting up the robot, can place trays anywhere, not rigidly in one place. The machine vision is kicking in, the red circles are detected wells. The user interface is being used. You can use the same equipment you would use in any lab. And it just goes through its paces. You don't have to program, you don't have to intervene. Same standard tips. The vision, again, the camera, you can see the camera there, the black little thing. And, ejecting pipette tips, the progress of the protocol as a graph, you can see that. And if there's a problem, the vision can detect the problem and let you know. It's very simple. Okay. If Google can build a car, this is much easier, believe me. So it's very easy to use, pretty accurate, and using advanced sensing, not just vision. So the main goal is to let you do science. That is our main goal. Okay? We do not want you to do programming and not do tedious manual work. So that is our main goal at A-BioBot. We believe there's a big untapped market. Alone in the United States, there are about 600,000 diagnostic testing labs, 200,000 academic research labs, mostly funded by NIH, if you count other agencies, many more, and several hackerspaces. We believe there's a huge market, especially if you consider the globe. We believe that the needs of this segment are not being met. Most folks in the lab require a lot of flexibility and a modicum of throughput. The current solutions offer a lot of throughput, but very little flexibility. A-BioBot, our platforms, will offer a lot of flexibility and sufficient throughput. So we have some early adopters. We reached out to our colleagues at Stanford and at Ohio State, and they've agreed to test a variety of protocols on our platforms. We also entered into some partnerships. We have a contract with Haliot for our systems, and we are collaborating with Arcturus BioCloud, one of our peer companies, and Modular Science to build a larger lab bench protocol, a lab bench platform. What we have right now is a desktop platform. We would wish to make it larger, and machine vision is going to play a very big role there. Also, we've been invited to install our systems in Beijing. COFCO has a very large installation. They invited us to come and try our system out. We estimate our cost to be $10,000, mostly for production, rapid prototyping. The value added is going to be through vision, software, user interfaces. These are our milestones. We already completed a prototype of our desktop model. Come summer, we would finish our lab bench model. Next year, we plan to ship out completely tested many protocols, as many as possible, because we want them to be useful for biologists, not just for engineers like us. So we believe that's the real goal, make them useful for biologists. And thank you very much. And we have the robot there. Some of you already seen it. Please come and see it again. And we welcome any comments and collaborations. And we are seeking to raise $1 million. Thank you.
Hello, everybody. My name is Adham Aljami, and I'm the CEO of Blue Turtle Biotechnology, where we're creating the next generation of therapeutics. Sometimes the smallest thing can have the largest impact. Take this healthy human cell, for example, with a functional enzyme, and compare it to one with a missing, depleted, or dysfunctional enzyme. Notice the buildup of substrates that enlarge the cell. Now, this is at the cellular level. What happens when you take a step back and look at the rest of the body? Well, because of one missing enzyme, somebody's diet can change, like lactose intolerance. Or they may end up with brain disorders, like Lendo-Kleffner disorder, and some don't make it until their 16th birthday, like Niehan disease. Now, pharmaceutical companies have been addressing this problem for years. They start by building small molecules of an enzyme in a laboratory, mass producing it in a facility, and then administering it to patients in a hospital. Now, the problem is, this is a very long and strenuous process. And along the way, we see errors sometimes. Because it's made in a laboratory, we have unanticipated toxic effects. Because of the heavy costs in these facilities, the patients end up paying the final price, sometimes being charged over $300,000 a year. And because some require hospital visits twice a month for the rest of their life, this isn't only a burden on the nation's healthcare, but a burden to the quality of life of these individuals. So at Blue Turtle, we decided, why not put this whole process into one factory, one that already lives in your gut. In a gut bacteria, you can make, mass produce, and deliver the enzyme right there in your body. The process is simple. We identify a missing enzyme, encode it into a vector, put the vector into a bacteria and grow it, put the bacteria into a pill, which makes it accessible not only to people here in the United States, but people all over the world, including those in developing countries. Give that to a patient, and once it's ingested, the bacteria will colonize in its natural habitat, and our engineered protein will be able to pass the gut barrier and into the bloodstream. This opens up a wide range of diseases we can target. In fact, we've identified over 500 enzyme deficiency diseases. But where do we begin? Well, 40 of those are considered lysosome storage diseases, and they affect about 200,000 people worldwide. These are all considered rare diseases, and the most common of those is Gaucher disease. We chose this as our initial proof of concept and go-to-market strategy for 3 reasons. 1, we can compare ourselves to current treatment and exceed it. 2, because it's a rare disease, we can file for orphan drug status, which shortens the time as well as the expenses to get to market. And lastly, it's got a sizable market. In fact, it's expected to reach $4.5 billion globally by next year, with the US market at almost $1 billion. So you might imagine a lot of companies like Pfizer, Shire, or Genzyme are targeting this rare disease. But what do they offer? Well, the first and most obvious is enzyme replacement therapy. You replace the enzyme, and it's been shown to be effective. But unfortunately, the quality of life for patients is low because they require hospital visits twice a month for the rest of their lives, and the cost of treatment is over $300,000. So what one company decided to do is, why not just reduce the substrates instead of replace the enzyme? It still prevents the cell from enlarging. So Genzyme released an oral pill. The enzyme doesn't get replaced, but it's shown to be somewhat effective. The quality of life for patients increases dramatically because there's an oral application, but the cost is even higher than enzyme replacement therapy. With a gut microbe, you're able to hit every single one of these at a much lower cost. But how much lower can we really be? Well, as I've stated over and over again, the cost for Gaucher treatment is over $300,000 per patient per year. The cost for us to produce this pill will cost us $300, which allows a very large margin and allows us not only to compete competitively in the US, but the rest of the world, including offer affordable healthcare in those developing countries. Now, as Arvind has said earlier, science is moving faster than Moore's Law. And in the last decade, the number of publications of gut microbes has increased tenfold. So we're seeing a lot of people in this space. Last year, a professor at Rice University got a 3-year grant to work on what they call the combination of the 2 hottest fields in life sciences, synthetic biology and microbiomics. And this year, a Belgian company called Ectogenics, which has been around for a couple of years, just got acquired for engineering gut bacteria for gut diseases. And this past month, a few Pfizer executives decided to launch Synologic, a company that engineers gut bacteria for gut diseases. Now, unlike any of these, we're going beyond the gut. So where are we now? Well, we incorporated this year, began the IndieBio program in March, and this week we partnered with one of the top CROs in the nation to run a small mouse study to show those of you that are skeptical that it does get past the gut. We look to expand the disease pipeline in November and push Gaucher to clinical trials by the end of next year, getting us to market for Gaucher by 2018. The team includes Nilesh Joshi and I as the co-founders, both with experience in multiple startups, both recipients of the Adams Entrepreneurial Fellowship, and both graduates of Wayne State, as well as a recent addition, Dr. Kengming Chang, a graduate from Johns Hopkins with experience at the NIH and Halliburton Energy. The ask? Well, I'm asking you to trust your gut. We're looking for partners interested in working with us for Gaucher, as well as other diseases, investors that want to help us accelerate both our research and help us build a strong company, as well as mentors and advisors who could help make introductions and guide us as we tackle this strong problem. So if you're interested in creating safe, effective, and affordable healthcare for everybody across the world, please see me after the presentations. Thank you very much.
Good afternoon, everybody. My name is Martin, and I'm the CEO of Xdem. And our team is creating the first population-scale stem cell library for organ and drug engineering. So we all came from stem cells, and at one point we were just a cluster of cells dividing and dividing before we even gained consciousness. Stem cells not only form the different tissues in our bodies, we also retain them in adulthood, and they regenerate your old or damaged tissues. These are the cells responsible for repairing your broken bones, your torn muscles, and even cuts on your skin. The photos that you see here are some of the tissues that we've already been able to create in our lab, including bones, knees, and even fat. However, stem cells are a very rare population of cells. Scientists can only find one stem cell for every 1 million cells of the body. But despite this, groups have been able to use stem cells to create lab replacement organs. So they've allowed us to develop better drugs using lab-made human tissues that's a step ahead of animal testing. And with their natural ability to transform into various tissue types and organs, stem cells really are the key to the future of drug development and regenerative medicine. But we're facing a stem cell crisis that's persisted over the last 40 years. It's incredibly difficult to isolate and grow these cells inside of the body. And the process of isolating cells from a donor is very invasive and involves drilling a hole into the hip bone just to get the raw cells. Ultimately, what this means is that there's not enough cells to go around for research studies. And because of this process, research is severely lacking in donor diversity. On average, scientists have access to only 2 donors for their studies. So groups have been extrapolating results from 2 donors to the entire population. If we want personalized medicine to be the norm, we need to be able to understand the differences between many people, and not just 2. And scientists are aware, and they acknowledge this flaw, and they know that there's a short supply of stem cells, not just in amount, but also in diversity. The groups that we've talked to have told us they have to go through 2 or 3 different companies just to get enough cells, and in the end, they don't even get the donor cells that they want. So they're essentially forced to purchase whatever they can take. But there's been an increasing efforts in the scientific community to get more diverse donor populations. They want to obtain more representative results for the population. And despite these challenges, the market for stem cells continues to grow exponentially. It's a $23 billion market today and expected to grow 4 times to $90 billion by 2020. Xdem started while I was working as a researcher at Columbia University, where I experienced firsthand the difficulty, complications with isolating and growing stem cells. Shortly after, I came up with the first prototype of the technology and generated stem cells from mice. I convinced Larson to join the team, And together we tested our systems on pigs. Nathan was there to build our devices and also program our learning algorithms. Alec was our first investor, and he's from Harvard Business School, and since then he's helped us with the early business development and strategy. Our technology platform now allows us to generate 150 times more cells than anyone can. And with this, we're creating the world's largest donor bank. And we're also using machine learning for the first time to predict stem cell behavior and how they turn to different tissues of the body. Our process for manufacturing stem cells is distilled into 4 simple steps. We collect the raw cells, we purify them into our process, we expand them over a number of days, then we package them to ship to our customers, or we bank them. And we have IP protection in the first 3 steps of this process. And with the technology we're building the largest donor bank for stem cells with over 10,000 donors. To put this into context, our nearest competitor has less than 10. Now we're doing this so that we can enable scientists to extend their models to the entire population. So now they don't have to rely on results from just 2 donors. And using this library, we're generating data for therapeutic modeling. We want to map connections between personal traits, like your age, your ethnicity, your exome sequences, to how well your tissues respond to injury or how well they react to different drugs. Ultimately, we want to create the future for ultra-personalized medicine. For example, today, there's over 10 different drugs a person can take for high cholesterol, but the doctor often does not know which one will work best for them. Using your stem cells, we can create your tissues in our lab, and with the help of your physician, test in parallel the 10 different drugs so that he can prescribe you with the most effective drug without you ever having to risk the side effects. We expect to generate $105 million in the next 5 years with 3,500 customers, each with a $30,000 customer yearly value. We're currently selling the best stem cells in the market, and there's currently no metric where we don't outperform the competition. For the first time, we're combining analytics for our stem cells and providing scientists with the largest selection of donors, over 10,000. And we know that our product is superior to our competitors. It's validated by our customers. And all this at a cheaper price than our 2 biggest competitors. Our first customer has told us that our cells grew very quickly and that they were very impressed with the differentiation and that they're the best cells that they've ever worked with in their lab. And we're currently generating revenue from our customers. We have a master services agreement with UCSF, and our product is currently being used and tested at the NIH, the National Cancer Institute, UC Davis, just to name a few. We began our process for human stem cell generation here at IndieBio last February. We got our first customer last month. We expect to be profitable in the next 3 months, and we're building a donor stem cell bank over 124 donors by the end of the year. And we're planning to launch the first version of the predictive software for stem cells by March 2016. We're here to look for investors to top up a round of $1.2 million and help us develop a stem cell analytics platform and to scale up to a truly global stem cell library. Thank you very much.
Taking a minute to get loaded up. All right, here we are. Okay. Hi, I'm Auryn BioCloud. I'm the CEO of Ophidia, and I'm here today to tell you how you can know your health now. The medical system is full of dedicated individuals, but the system as a whole isn't working. Consider when we have a stomach ache. Maybe we think we should get tested because it could be something serious wrong with us. But usually we don't, because we know that if we go to the doctor, we'll have to book an appointment, we'll have to go there, we have to wait to be tested, and we have to wait for our results. We have to have a lot of blood taken, and we have to pay for this whole process. So we delay, we don't get tested, and usually that's fine. But sometimes it's not, because we could have had stomach cancer. And in fact, this is not just an isolated incident. This adds up to a gigantic tragedy today. 1 in 3 people who die of disease could actually be treated if their disease was diagnosed earlier. It's a strange world that we live in, a world where we have access to so much data through our phone every second, and yet we don't have access to the most important data of all, our own inner data. This is why I founded Ophidia, to transform healthcare today. And I brought together a great team: electronics, mechanical engineering, proteomics, and microfluidic technology. A really experienced and talented and awesome team to create the Ophidia solution. The first system able to do 50 tests in a portable device at lab accuracy from 1 drop of blood. And here it is. This is our prototype, and I'm unveiling it today. The first time we're able to see our Ophidia solution. To make this, we created new technologies around new microfluidic components, new surface chemistry, new bonding techniques at the microscale, and novel assay control. This allowed us to file 5 patents on our core IP and create the highest precision system at the microscale ever created, which means for the first time we have a portable system that is able to compete with the lab in accuracy and breadth of tests. And now, for the first time, we're going to see this happen in public. We're going to see this being used. And my co-founder here, Jake, is going to take this chip, and he's going to put it in, and we're going to see a blood sample tested right here. Put it in. Put it in, Jake. It's going to happen right now. So, great. It's going in. Bear with. Okay. Oh, go and get the other chip then. Okay. So, demo is failed, but we're going to put it in, and you guys come over there to the other side, and you're going to see it. And if you're interested in the results, then you're going to see that being happening right now. And we can unveil this blood sample being tested right there. And we'll be happy to show you the results straight off of this presentation. So, our system works like this. You put a blood sample onto the chip, and it goes into our reader. And our reader can be placed in any of these locations, in primary care clinics, urgent care clinics, pharmacies, any location, which means that we are 1 block away from where everybody is, so that people can just go 1 block from their home and be tested straight away. And those results will be available to patients immediately, but also to physicians. So the physicians will be able to access data and be able to learn how to be able to more effectively treat their patients. And we'll create tools to enable them to do that. So why is Ophidia needed? Ophidia is a new type of diagnostic system. Traditional point of care has been low accuracy and a limited number of tests. Lab-based systems are slow, they require a lot of blood. Ophidia is a new generation of point of care, 200 times faster than lab-based systems, but would also enable us to do 10 times more tests than traditional point of care systems. And it will work from 1 single drop at lab accuracy. Our business model is straightforward. We sell to clinics, B2B. It's a razor blade model. We're able to make these readers at 1K, and each of these chips are only $5, which means it's only 10 cents per test to make. And we're going to sell these at $20 to $100, meaning we give recurring revenues per patient. And we're giving away the reader so that we will be able to get this out to as many places as possible. we're building great clinical partnerships with partners such as CVS, Kaiser, Halyard, and Brown & Tolland, which will mean that we'll be able to reach hundreds of thousands of doctors and millions of patients around the world. Our regulatory process is a 510K. All of these tests have predicates. This means that we'll be able to launch our product within 2 years by late 2017. We're aiming to do all of these most common tests. These are the tests that all of us will need many times in our lifetime. And we'll also be able to do extra types of tests, such as cancer biomarkers. Many of these have never been done at point of care before. So we're here right now. We've filed our core IP. We have our system demo working, which is lab accuracy with 2 tests. And this puts us already ahead of these key competitors, such as Lear, Roche, and Johnson & Johnson. We're raising our seed financing now. And this will put us forward to go through QA, manufacturing scale-up, and be able to then go through the approval process working with our great clinical partners to launch our product by 2017. So if you're interested to find out more and learn how you can know your health now, please come and find us afterwards. Thank you.
So that was 12 companies. What do you guys think? That is pretty amazing, guys. I think having all of them working together, it was amazing to me to watch 30 some odd people come together and just really gel as a team, bigger team, and then kind of as a family. And the first batch really did feel like a family. So that I always am really excited about that, and really, again, glad to be a part of it. Also, can I get the Ron, Ryan, and Manana to come up here, wherever you guys are? Come on up. So, because without these guys, none of this would happen. I want to also introduce Manana, Miss Ropian. Thank you. Manana is the general manager of Where We Are, and Where We Are is the revolution. So this is our new headquarters for SOSV here in San Francisco. Downstairs, we're going to be building a much bigger lab than we have currently in the dog patch. Biosafety level 2 lab, and the future batches will be run out of this location. Upstairs, we're going to be having co-working spaces for both portfolio companies, but for companies that are really trying to change the world in a significant way and make the impossible possible. That's really important to us, and so we'll be offering it at a lower the market rate just to support these teams. Yes, Ryan, do you want to say something?
I was just going to say, and we are currently taking applications for our next batch, which is $250K in funding for each company. It's a $250K package, and early application deadlines are the end of June.
So in like 2.5 weeks, 3 weeks, the deadline. So yeah, I mean, we're really amazed by the success of the experiment, which is batch 1. Really, truly an experiment to see how this would go, and beyond our expectations. So we're extending the runway for all the teams that are coming in. I also want to bring in Sean O'Sullivan, Mr. SOS of SOSV. It's always remarkable for me to meet someone who's inspiring and someone who actually believes enough in a future that could happen to be able to underwrite and fund this and create something as special as the organization SOSV. So I'd like you to put you on the spot and say a couple things.
Well, thanks. I just, I'm an engineer, so all this science is actually quite confounding to me. I like to build, hardware and software and things. And so when people talk about, creating life forms and all that, it just seems like it's a real stretch for me, to comprehend what's going on. but it's so incredible. And I can say, in my own life, my own mother had diabetes. And so the first real, great synthetic biology drug that came out was insulin. She used to take insulin that was made by cows and pigs. That's how they used to do it. And so Genentech came out with a synthetic biology product for human insulin. It cost hundreds and hundreds of millions of dollars for them to create that back in 1982 and release it to the marketplace. And that actually made insulin treatment much more available for many people and much more safe as well. So the things which used to be inaccessible or cost humongous amounts of money, like sequencing the human genome, which was done in 2000, cost $1 billion. Now you can get it done for $1,000. What we're doing here with IndieBio is actually quite, I don't know, it may seem like it's pretty impressive. It's remarkable, actually. It's the beginning of a new era where we can get these companies launched, where we're democratizing the capability of creating the science and making life-creating, life-affirming new offerings available to billions of people on the planet. It's I know the market numbers. It all seems like it's about the money, the billions of dollars, et cetera. But a lot of us have mothers and sisters and brothers that are affected by these diseases. All of us are affected by the overuse of resources on the planet. These are so critical to our quality of life as human beings that I urge all of you that believe in any of these startups to take out your checkbooks and help make their outcomes possible. We can only guide them a certain part of the way, but we do think that there are commercial opportunities. But it's so important for the planet that these technologies hit the market. That I do hope that you all realize what you've seen today is actually incredible. Actually, it's really a testament to these guys and these gals. And so many of the people, the founders, of course, that have made this such an exciting and incredible experience. I've learned so much to be part of this, and I hope you've learned some incredible things from the presentations you've seen today. Thank you very much for coming, and the teams will be on the side, and I guess there's an open bar and stuff downstairs. Yeah. So you'll
actually, Manana, you want to mention this? No. Come on, Manana. You have to come.
Tell us where the catering is and all that other stuff.
All right. So we actually have catering upstairs and downstairs, and there'll be an after party for people that like to dance. Yeah. No after party. Drinks, and yeah, it'll be an open bar.
Awesome. So, thank you all for coming, and I hope to see you soon. Thank you. Bye.